Make sure that we get this recorded. Okay, missed a couple questions, but that's okay. So yeah, so if it talks about how many are produced or how many total ATP are produced, we want to say that there are 4 ATP that are produced during glycolysis. If it's asking about the net total of ATP, because we need to use 2 ATP in order to make, we use 2 ATP in order to create those 4 ATP, our net total is 2. So just be very, very careful on this wording. This is why I put this question here, because you most likely will have one or both ways of asking this question. And if you read the question correctly, it's free points. Let's go. How many ATP are produced during the Krebs cycle from one glucose molecule? So this is now just talking about within the Krebs cycle from one glucose molecule, how much ATP or GTP gets produced. So a little bit of confusion on this one. So in this difference here now, we're no longer talking about glycolysis, we're talking about the Krebs cycle. This is the next step. So we have glycolysis, and this is a big thing to keep each of these. They're all linked together, but each of them is a separate process. Okay, glycolysis happens first within the cytosol, then we're going to have the Krebs cycle happen. And this right here is our Krebs cycle. Let me clean it up.
I actually have a question.
Yeah, what's up?
Because I know in his book it says that 2.5 ATP are made from 1 NADH, and there's at least 3 NADHs, so that's like 4, 5, 7.5 ATP. Or am I just thinking of that wrong, or no?
So you're thinking of it along the right lines, because that is true. So the NADH and the FADH2 are going to be used a little bit further down the chain to make ATP. So right now we're making NADH, we're making FADH2, and if you remember, also in glycolysis, we actually also make NADH. Remember, right here, we make 2 NADH in step 6 of the glyceraldehyde 3-phosphate to the 1,3-bisphosphoglycerate. So this NADH is eventually going to be used to generate ATP. However, right now we're just making the NADH in glycolysis in the Krebs cycle. We make the ADH, but it does not yet turn into ATP. It will continue, that the NADH will actually continue and go into the electron transport chain. And that during the electron transport chain is when it will get converted to ATP. So for this question, specifically asking how many are produced during the Krebs cycle from one glucose molecule, each turn of the Krebs cycle is going to produce 1 ATP. And 1 glucose produces 2 acetyl-CoA, so we get 2 turns of the citric acid cycle. So in the Krebs cycle itself, from 1 glucose, we get 2 ATP. Then we also get the potential ATP from the NADH and the FADH, which will get converted later on. Does that answer your question, Christian?
Yeah, understood. Thank you.
Awesome. Yeah. Becca's still in the lead. Okay, so which of the following steps does not produce NADH? This is kind of getting a little bit deeper into the material. Good. So if anybody has not watched my YouTube video, I do go over a couple of things in there as ways to kind of help simplify and remember the Krebs cycle. So we have a nice little mnemonic down here. Couples cuddly insanely often after special sex for marriage obligations. The acronyms or like mnemonics like this are great ways to remember these because there are so many steps. Even if you can just remember the first letter, CCIOASSFMO, really, really helpful to help remember the order of all of our intermediates of the Krebs cycle. Because let's be honest, just straight memorizing this is not all that easy. The NADH, the mnemonic to remember this is I am the most important. So in these spots, the isocitrate, isocitrate to alpha ketoglutarate, the alpha ketoglutarate to succinyl CoA, I am, and then malate to oxaloacetate, the most important. Those are our three spaces where we produce NADH. Same thing with the citric acid cycle. You'll want to remember which locations you also produce FADH. And occasionally he will also ask which locations you produce CO2. It's good to try and draw correlations between this. So if we look at all of our different enzymes and our different reactions, we have these dehydrogenase enzymes specifically the isocitrate dehydrogenase and the alpha ketoglutarate dehydrogenase complex. We'll see where we get our CO2 that comes out. And then you'll want to know which step the ATP or the GTP is formed, as well as between which steps the FADH2 and the NADH are made. So there's a lot, like I said, I say this in the YouTube video that I recorded, there's a lot of different tidbits within this whole cycle that you need to remember where stuff happens. So if you can remember well the order of the intermediates first, which is that mnemonic, the couples cuddle insanely often after special sex for marriage obligations. If you can remember this is a good start, because then our next step are enzymes. Pretty much all of our enzymes are going to include the name of what they like the name between this, like acetyl-CoA to citrate is citrate synthase, and conatase makes our cis aconatate, and so on. So like the names of each of them are included. The enzyme includes whatever is before or after what is being either worked on or what is being produced. So like I said, if your guys' test is on Monday, you really, really, really want to spend some time going over these steps. Ideally, if at all possible, you want to be able to draw almost this entire thing, not like all of like the pictures and stuff, but the flow of it, where acetyl-CoA gets acted on by citrate synthase to produce citrate. Citrate gets acted on by a conatase to make cis-aconatate, and so on, and be able to do all that, and be able to put in where NADHs come off, where CO2s come off, where FADHs come off, and where ATP comes off, because that will be a good portion of questions from your test will be asking stuff like that. Becca, still crushing it. The Krebs cycle takes place in the cytosol, the interstitial fluid, the outer mitochondrial membrane, or the mitochondrial matrix. Great job. So cytosol, what other process happens in the cytosol?
Glycolysis.
Awesome. Glycolysis happens in the cytosol. The Krebs cycle happens within the mitochondrial matrix. And then next week, after this next exam, we'll start to talk about the electron transport chain. And that we'll actually look at happens in between the inner mitochondrial membrane. The mitochondria has an outer membrane and an inner membrane, an intra, I think it's an intramembranous space, and then the matrix being in the middle. So the Krebs cycle happens in the mitochondrial matrix. I promise you will either get this question on this test, or you will get where glycolysis takes place, or you will get both. Very, very common question that you'll be asked. During each cycle of the Krebs cycle, which of the following is produced? A little bit of confusion on this one. So if we think back real quick, the mnemonic that we just talked about for remembering where NADH comes off, can anybody recall what that mnemonic was? Anybody at all? Anybody recall what it is?
I am most important.
Good. I am the most important. So if we know that there are 3 NADHs, this question, we can start to pick apart the other answers. None of the other answer choices had 3 NADH. So we have 3 NADH, 1 FADH2, and 1 GTP. If I had changed the question, if I had asked during with the Krebs cycle, how much NADH is produced from 1 molecule of glucose, what would the answer be then?
Would it still be 3 NADH?
So this is during each cycle of the Krebs cycle, right? And how many cycles of the Krebs cycle do we get per glucose?
Oh, per glucose? 2.
Exactly. So we get 2 per glucose. So essentially we could take this and we could double these values. We'd have 6 NADH, 2 FADH2, and 2 GTP. I could also ask the question, how many NADH total is produced from 1 glucose after glycolysis and the Krebs cycle? Combine the 2 of them.
8.
Good. We would have 6 from the Krebs cycle and 2 NADH from glycolysis. What if I ask the question, how many NADH is produced from 1 glucose in glycolysis and the Krebs cycle in an anaerobic environment? This gets a little bit into the...
Is it still 2?
This one, yeah, that one would be 2. Why would it be 2 instead of 8?
Is because anaerobic, that's like no, that's I know it's no oxygen. Is that glycolysis or?
Exactly. So if there's no oxygen at the end of glycolysis, so the goal of glycolysis is to make pyruvate. And glycolysis will make pyruvate as long as there is oxygen present in an aerobic environment. But as soon as the oxygen goes away and there's no more oxygen left, we're in an anaerobic environment. Instead of making pyruvate, what gets made in glycolysis?
Lactic acid.
Exactly, lactic
Lactic.
acid. Yeah, so make sure you pay very, very close attention to aerobic versus anaerobic, because you are going to have questions where that is going to completely change the answer of the question. If it says aerobic, so if the question could ask in glycolysis, in an aerobic environment, how much lactic acid is produced during glycolysis? And the answer should be 0, because in an aerobic environment, there is oxygen present, so you get pyruvate. So those are a little bit more on like the tricksy side. Dr. Sarkar doesn't ask a lot of questions like that, but you will see something like that eventually on boards. So it's good to really understand aerobic versus anaerobic.
I got a quick question. For the Krebs cycle, like in total, like if we were to go through if we were to go through glycolysis and then Krebs cycle as well, how many total NADH would that be? So that's like two. That's 3 times 2 for Krebs cycle, so that's 6. And then one pyruvate, or it makes 4 in glycolysis, or just 2?
It makes 2, 1 per pyruvate that it's producing.
Oh, okay. Yes.
Yeah, so here's our, bam, right there's our glycolysis. So because we have 2 glyceraldehyde 3 phosphates, we make 2 NADH. So you get 2 NADH total from glycolysis, and then you end up with 6 from the Krebs cycle after both pyruvates have gone through. And this is just the matter of make sure you read the question and ask, because the way that he'll word it, he'll have questions that are worded per glucose molecule, or he'll have it per pyruvate or per acetyl-CoA, something like that. So just make sure you read the full question to understand what it's asking. Next, what is the first rate-limiting enzyme in glycolysis? Pyruvate kinase, PFK, hexokinase, or phosphoglycerate kinase? Good job. So remember that the good thing is if you picked PFK, PFK is going to actually be our second rate limiting enzyme in glycolysis. I think I have a question about this a little bit later on, but I'll show this right now. Remember, in glycolysis, or whenever we talk about regulatory enzymes, this is something that you are going to be asked about on exams. This is something that he loves to ask about because it is very, very important. It's very, very easy to ask these sort of questions. So we have hexokinase, phosphofructokinase, step 1, step 3, and then our pyruvate kinase as well, which is towards, I don't know, it's either step 9 or it's step 10. It depends on how you count it. Technically step 10 if you count the way you do in Dr. Sarkar's book. Going from phosphoenol pyruvate 2-pyruvate. Cool. Remember these, the rate limiting steps. He always has questions on the rate limiting steps. Most commonly, Dr. Sarkar likes to ask about PFK. That's one that he really, really likes. So for sure, make sure you remember that one. Which of the following is the 8th step of glycolysis? This one's a little bit mean, but you will have questions similar to this on the exam. And if it's something that you have, once the time runs out, we'll talk about kind of how we can go about answering this type of question if we realistically have no idea. Nice. Very good. So with this, it's great if we just have it like each step memorized. That is awesome. But if we want to break it down and think, okay, how can we go about this if, let's say, it asks about the 4th step or the 5th step and we really don't have any idea. So what we want to think of, the big picture ideas of glycolysis. So the very, very beginning, if we know the beginning, we know the end, right? So we know that there's 10 steps to glycolysis. And so we think pyruvate is the very, very last step. So phosphoenol pyruvate 2-pyruvate, that has to be our 10th step. So it can't be our 8th. The glyceraldehyde 3-phosphate, but we have that here twice. The dihydroxyacetone phosphate to glyceraldehyde 3-phosphate and glyceraldehyde 3-phosphate to 1,3-bis-phosphoglycerate. So if we can think and we can recall that glycolysis is split up into 2 different phases. Can anybody recall what the name of those 2 phases are that we split up glycolysis in?
Pre-phase and payoff phase.
Yeah, we have the pre-phase or the priming phase and then the payoff phase. So the priming phase is going to be the first couple of steps, and we think that glyceraldehyde 3-phosphate, you can think of the formation of glyceraldehyde 3-phosphate is our cutoff between those two. So if we remember that getting to that point from glucose to glucose 6-phosphate, fructose 6-phosphate, fructose 6,1,6-phosphate to our glyceraldehyde 3-phosphate and our DHAP, getting to this point right here is our is our priming phase, our preparatory phase. And then we convert that DHAP to a glyceraldehyde 3-phosphate, and thus begins our payoff phase. So that's really split right in the middle. That this getting to our second glyceraldehyde 3-phosphate is our step 5. So remember, that is our midpoint. So if you can remember glyceraldehyde 3-phosphate and dihydroxyacetone phosphate right there, getting to our glyceraldehyde 3-phosphate, this is our midpoint. So if you know the end and you know the middle and you know the beginning, you'll be in much better shape to figure out where everything else goes. So having those big main ideas, especially like if you're getting close to the test and really cramming, and you just cannot get this to fit in your brain, go a little bit more broad with it. That it goes glucose to fructose to the glyceraldehyde or to the G3P, and then it goes to all of its fun phosphoglycerate whatevers, and then it gets to pyruvate. So if you can't remember the name of each one specifically, get the general idea down, so the general order, to help yourself be able to figure out the questions in the middle of the test if you need to. How many pyruvate are produced from a molecule of glucose during anaerobic conditions? I got you guys. Okay, so who can tell me why the answer is 0?
Because in anaerobic conditions, it would be lactic acid.
Very good. These are the kind of things. So anaerobic, please make sure that you know and that you read this very carefully. If you ever see the word anaerobic or aerobic, read it super carefully and understand what those are. This is something that will not go away. Aerobic and anaerobic is something that gets covered in Biochem 1. You'll talk about it again in Biochem 2, in Phys 1, in Phys 2. You'll talk about it in systems pathology, in clinical nutrition, in physical diagnosis. You'll talk about aerobic versus anaerobic, even like in other classes as well, in PT and a couple of others, you'll have anaerobic versus aerobic questions come up. So this is a sticky point. A lot of people get these definitions mixed up. Anaerobic means without oxygen. There's no oxygen present. Aerobic means that.
Sorry.
Go ahead, Christian.
So there's no pyruvate created. The only time there's pyruvate is when it's going to like the Krebs cycle.
Exactly. So let's do this.
Where does it cut, like in glycolysis, to go to like the anaerobic route, lactic acid? Like, is there like a cutoff point in glycolysis where it's like?
Yeah. So let me pull this up real quick. So. It's also something I want to make sure that we. Okay, actually, I'm gonna clarify this a little bit because this is actually something I was a little bit off on. Okay, because technically pyruvate actually does get created, but it doesn't get converted to acetyl-CoA. So this is my mistake. Let me make sure that I correct this for you guys. I was going a little fast. Okay, so technically the answer to this question would be that 2-pyruvate are still produced. However, those pyruvate do not continue to acetyl-CoA. They get converted to lactate. This is my mistake. I'm sorry, guys. all in the process of learning. So
No worry. Do I still get my Kahoot points?
yeah, I'll make the edit and add your points in. I'll make sure to change that question as well. Kind of word it a little bit differently. So instead of pyruvate, it would be better saying how many acetyl-CoA are produced from one molecule of glucose during anaerobic conditions. Would be a better question. So this chart right here gives us the idea of what sort of happens. So glycolysis is going to be what we call a mixed process, meaning that it is both aerobic and anaerobic. He can ask this sort of question as well on the test. So glycolysis can be both, or it's both. In either condition, it's going to continue to happen. Glycolysis will happen in aerobic or anaerobic conditions. When we get down to the Krebs cycle, this is a citric acid cycle. This is aerobic only. Is a citric acid cycle. So, and when we do have oxygen, with oxygen, our pyruvate gets converted to acetyl-CoA and will go into the citric acid cycle. But when we do not have oxygen, our pyruvate gets converted then to lactate, aka lactic acid. And then he could also ask you a question about this, specifically talking about yeast, where our pyruvate gets converted to alcohol and carbon dioxide. I do remember him having a couple quiz questions and exam questions just about this little piece right here. So I would remember this. So I'm gonna give me 1 sec. I don't know if you can hear my cat screaming at me in the background. Okay. So this little chart here, glycolysis is a mixed aerobic and or anaerobic. We get pyruvate with oxygen. We get continuing into the citric acid cycle, and then eventually down into the electron transport chain. When there's no oxygen, we get lactate, and whenever we have yeast or certain bacteria, we get alcohol and carbon dioxide getting produced. This is actually the way that they make wine, or what wine or beer, most alcohols, is from this process. Okay, that's a good clarification. Does that make sense to everybody now? I want to make sure that's clear. I apologize again for my mix up.
Yes, all good.
Okay, awesome. True or false? Glucokinase is found exclusively in the liver. This is true. So glucokinase, or actually, is there something that I am missing as to why it would be false? I want to make sure that I'm not missing something.
No, I think you're good because hexokinase is not in the liver, right?
Hexokinase is in the liver still. However, so hexokinase is found in all of our tissues, in our muscles, in our kidneys, and like all throughout the entire body. However, glucokinase is going to be found in the hepatocytes in the liver. So the liver cells have an extra one, which is glucokinase. The reason being is that the liver is a little bit different in its function with glucose, because even though even after it brings glucose in, the liver is still able to release glucose back into the blood. So glucokinase and hexokinase do similar jobs. They do it slightly differently to allow for the release again of glucose back into the bloodstream from the liver. We're not that's as deep as we're gonna go into that, just because if we get any deeper, that's cutting into some stuff that you'll learn later on, and it's a little bit above y'all's pay grade right now, so don't worry too much about that. Just understand that the glucokinase is concentrated in the liver, and hexokinase is found in all of the cells. CC coming up. Dihydroxyacetone phosphate to glyceraldehyde 3-phosphate is what type of reaction? Good job. This one was a little bit harder, a little bit more niche. So really good job, though. I'm glad that majority of people got this one. So these are a couple of other questions, the type of questions that you can be asked. The trick to a lot of this, I'd say, is to try and recall the names of the enzymes. So the triose phosphate isomerase is the enzyme that acts on our dihydroxyacetone phosphate. So it's in our isomerase reaction. I'm glad that nobody picked the mutase, because that's where most people tend to get mixed up. So with this, actually, can somebody, does anybody know what the difference is between an isomerase and a mutase enzyme? What the difference between the two of those are or is?
Like what it does in glycolysis, or like its like actual definition?
It's actual definition, like what an isomerase enzyme does that's different from a mutase enzyme.
Does it I mean, I can take a shot at it, but
Go for it.
well, I know that it mutase, it just moves the phosphate over. So maybe mutase is just the moving of a phosphate, and isomerization is just restructuring of the same elements, basically. Or
Very, 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 very close. So mutase are going to move a functional group like a phosphate. So the mutase is going to move a functional group within the same molecule, whereas an isomerase is going to rearrange the entire structure of it. So very, very, very good, Christian. That's awesome. So mutase and isomerase, they're very, very similar to each other, just knowing that a mutase just changes the location of one functional group, whereas the isomerase will actually rearrange the whole structure of can rearrange the whole structure of the molecule. Very good. If you read this question the first time, you're like, oh crap, make sure that you go back through and review this whole part of the book. This whole part of the book that walks you through each step of glycolysis and of the Krebs cycle is really, really, really good, because it goes through exactly what's happening. It tells you what type of reaction that there is, and these sort of definitions, the things that that you see here, is where he'll pull a decent amount of questions from this type of stuff. If anything, if you're able to read through this and take your time to understand what the words are and like recognize what the words are saying or what they mean, when you get to the test, reading the questions will be much easier. If it's the first time that you've seen some of these words or some of the different, like the different names of the enzymes or the different intermediates, it can be really, really stressful if you haven't seen those before. And it just makes the test a little bit harder. Whereas if you've seen a lot of the words before, it's much easier to get through it and to understand what the questions are asking. Becca, back on top. Okay. Glucose, amino acids, and fatty acids all can be converted to which of the following? Good job. Here's our little chart. All of our stuff. Wrong one. all of our stuff. It comes back and we can bring it into acetyl-CoA to enter into the citric acid cycle. Using our fatty acids and amino acids as energy source happens when we run out of glucose, which there's a special name for it, which I have another question on that a little bit later on. Okay, pyruvate dehydrogenase complex. Which of the following is not a coenzyme used by PDC, or the pyruvate dehydrogenase complex? That's kind of what I expect with this. So the pyruvate dehydrogenase complex, you should not have a ton of questions covering this because he does not really focus on it a lot in the lecture material, but he does have a substantial chunk in the actual reading that covers it. So the job of the pyruvate dehydrogenase complex is the actual converting of pyruvate into our acetyl-CoA. And this is why right here, we need oxygen because we get the production of CO2. So oxygen also has to be going in to it in order for this dehydrogenase complex to work. And there's 3 different parts to this complex. And they call them E1, E2, and E3. And there's 5 different coenzymes that are used. TPP, FAD, CoAsh, NAD+, and lipoate. It's obviously it's been a while since I took Dr. Sarkar's class, but I do recall quite a bit about TPP. When I saw that again, I was like, for some reason that pulls up my memory strings. TPP being something important and something to remember. So whether or not he still tests on it or why exactly I have a little bit of a nightmare memory of TPP, I don't know, but takes that with a grain of salt. That more than likely, if just from reading through this section, I feel like more than likely he'll ask questions, asking about the coenzymes, or potentially the functions of the mechanisms of action of PDC. So this part right here. So if you haven't had a chance to review this, I go back through and review this again. I cover all of this in the YouTube video as well. So if you want to go watch that as well, it's good just to, I'd recommend if you don't have a lot of time to study, you can always, with those YouTube videos and stuff, throw them on either like in the car or like when you're doing other things, or Ninja Nerd videos or something, anything you can put on in the background just to see this material a couple more times. Okay, so PDC is inactivated by all of the following except. So remember, I'm going to talk a little bit about this. So remember that PDC is converting pyruvate to acetyl-CoA. So think about in what situation would we not want to convert more pyruvate to acetyl-CoA? Okay. This is another one. So when we read these questions, these are the ones when they say except, a lot of the times these are the ones that get a lot of people as well, because they'll read the PDC is inactivated by all of the following and then go down for the choice, but they miss the except. So PDC, if we look at it again here, the pyruvate dehydrogenase complex is converting pyruvate to acetyl-CoA. So we have what's called a negative feedback loop, meaning that once we have certain products that are produced and that are then readily available, they're going to kind of feed back or pass the information back into the system saying, hey, we've got what we need. We don't need to keep making more. So once in the Krebs cycle, or let's say in the Krebs cycle, in the Krebs cycle, once we produce enough NADH to be passed along, the NADH is going to get registered back by the PDC, and it's going to say, okay, we have enough. We don't need to keep converting pyruvate to acetyl-CoA. If we have a bunch of acetyl-CoA floating around, so okay, we have a bunch of acetyl-CoA, which means it's not being used up in the Krebs cycle, which means that we have enough. We have sufficient for what we need. And the entire goal and the focus of this whole process is to produce ATP. So once we get enough ATP produced, that's going to give negative feedback to the PDC and tell it to shut off, and then it no longer needs to keep making more. However, if there is pyruvate is one of the substrates, one of the reactants for PDC. So if we have a whole bunch of a big buildup of pyruvate and we don't have acetyl-CoA, don't have ATP, and we don't have NADH, then the pyruvate will push the PDC to be activated and to continue to produce and continue to convert pyruvate to acetyl-CoA. Creation of glucose from non-carbohydrate sources is known as what? Oh, I'm so proud of you guys. Thank you for getting that right. That is awesome. This is another one. He'll give you questions like this. He loves these questions where all the answer choices will be your different glyco and glucose, and it's just a straight definition. So just make sure you know all of those different glyco and gluco words, as he'll ask questions on those. Malate to oxaloacetate is what kind of reaction? Dehydrogenase, hydration, dehydration, or oxidative decarboxylation? Ooh, my bad. Good work. This is more of just kind of our straight memorization and practicing, where we have all of our different reactions: condensation, dehydration, hydration, oxidative decarboxylation, oxidative decarboxylation, phosphorylation, dehydration, dehydrogenation, hydration, and then a dehydrogenation as well. So just draw this out a couple of times. If you can, guys, I would sit down tonight and write this out 2 or 3 times. And then tomorrow, take out a chunk of time and do the same. And then the same thing on Friday, and then on Saturday, and then on Sunday. And if you do it 2 or 3 times and try and do as much of it from memory as you can, and then come back and check yourself and then correct yourself, I promise you, you will feel so much more confident going into your exam. How many net ATP are produced from glycolysis up to the end of the Krebs cycle for 1 molecule of glucose? Okay, so these sort of questions, make sure you guys come back and look at these again, and break them down and understand these. I would make sure you practice these until you really understand them. So we talk about net ATP from glycolysis all the way to the end of the Krebs cycle. Remember, we talked about each glucose in glycolysis. What is our net ATP from glycolysis for 1 molecule of glucose?
Two.
We get 2 is our net from glycolysis, because we create 4 and we use 2. And for the Krebs cycle, each turn of the Krebs cycle, how much ATP or GTP, same thing, is produced for each turn of the Krebs cycle?
Each turn is 1, and you do 2 turns, so you get 2.
Perfect. And the 2 plus 2, we end up with 4. Good. Please, I promise you guys, he's gonna have a couple questions like this. So hopefully, if you're struggling at all with this concept, spend some time to reread these questions. Go back. I will. This video is recorded, and I'll also go ahead and download it and post it to my YouTube account, YouTube channel, so you guys can come back through and look at these questions, stare at them for a couple of minutes, read them really, really carefully, because if you understand it now, when you get to your test, hopefully these will all be free point questions. Which of the following is not considered a rate regulating enzyme of the citric acid cycle? I gotcha. Okay. So the regulatory enzymes for the Krebs cycle. Little mnemonic for you. The CIA is always watching. The CIA. So our citrate synthase, isocitrate dehydrogenase, and the alpha ketoglutarate dehydrogenase complex. Again, this is just another little memorization thing. hopefully keep this in mind. The CIA is always watching.
Wait, what was the question again?
Which of the following is not considered
Right?
a rate regulating enzyme of the citric acid cycle?
Shouldn't it be
Oh, hold
succinate?
up. Yeah, it should be. My bad. Where I misclicked it when I was setting it up. My bad. It should have been
Okay.
succinate dehydrogenase. I'll go back and fix that. My baby was screaming for 3 hours last night. I don't know what's going on with my brain. Dehydrogenase, succinate dehydrogenase. I'll make sure to go back and fix that. So whoever got that one right, good job. I know the scores are a little bit messed up. Sorry, guys. So following the path of the citric acid cycle, succinate gets turned into what? Good job. Remember mnemonic? Couples cuddle insanely often after special sex for marriage obligations. Question 21. How many ATP are formed per NADH? Very good. How many ATP are formed per FADH2?
$1.5.
Very good. This is something that, for the purposes of this class, remember 2.5 and 1.5 for those values. Later on in boards, it's really kind of annoying because occasionally boards will use the values 3 and 1, 3 for NADH and 1 for FADH2. But the most accepted is 2.5 and 1.5. So as for this class, use 2.5, 1.5. Or sometimes like the math won't work out perfectly. And if that's the case, just pick the closest one. He won't ask you, he won't give you a number that's a ridiculous amount far off. So if we do some quick math here and we stop and we think about it, with the amount of NADH and FADH and ATP that we produce, if it runs all the way to the very end, roughly how much ATP do we get per glucose molecule?
I don't know what I'm counting it wrong, but I'm getting 21. I don't think that's right.
I got 11 and a half.
So things so all the way through. So per glucose, let's do all the math. All right, so we have one glucose. And then we have glycolysis. Within glycolysis, we get 2 net ATP. It's doing that, it's weird. Whatever. Then we have the Krebs cycle. We get another 2 ATP. We also get 2 NADH. And how many NADH do we get from the Krebs cycle per glucose molecule?
We get 6 or 3 at each turn, right?
3 each turn, but 6 total. And then we get 2 FADH2. So 2 NADH times 2, that gives us 5. There's 2 here, 2 there. 6 times 2.5. 6 times 2 is 12. 12 plus 3 is 15. And then another 3 here. So 5, that's 20, 22, 23, 24, 25, 26, 27 is what has us up. Let me find, I might be missing one because I know they range at 30 to 32. Here we go. So here's all of our beautiful math. Okay, so we lose 2 ATP, so we have -2. The number of ATP, so we get the 2 NADH from glycolysis plus the 4 ATP from the rest of glycolysis. Then we get NADHs 2, 1, 2, 3, 4, 5, 6. Hold up. This chart is different than what it's supposed to be. Where's the? I never noticed that. I did not notice that before. Because we should have 1, 2, 3 NADH here. Yeah, so give or take about 27 or so net ATP is what we should have. I'll have to ask Dr. Sarkar about that chart. So when we do all the math works out to 27. The way that he'll give you the question, if you follow the math like this, knowing that the 2 net ATP, 2 NADH, 2 ATP, 6 NADH, and 2 FADH2, you should get the correct number. So we'll say it should be about 27 net ATP. It won't always, the answer won't always be 27. It might be 28, 29, something like that. Just depends on what values that you use for the NADH. Okay, last 3. Citrate can be used to form sterols. True? All good. True or false? Glutamate can be derived from alpha glutarate from the Krebs cycle.
Can we talk about that sterile question? I have no clue what that is. I guessed.
Yeah.
And this one.
Yeah. After this last one, there's a slide that goes over this. These last 3 questions are all kind of linked together. Oh, I must have, the timing didn't get saved on these ones. Okay. So all of these questions come from this chart right here. So this is kind of talking about how with the citric acid cycle, all of our different intermediates can be used for other purposes besides just the Krebs cycle, besides just producing NADH and just producing ATP. So citrate, for example, can be turned into or be used to create fatty acids and sterols. Sterols is something that you guys will actually talk about next trimester in Biochem 2. A sterol is a type of chemical compound For example, cholesterol is a sterol. Steroids are sterols. Like testosterone, estrogen, those sort of things are going to be our sterols. Alpha ketoglutarate can be broken down to produce glutamate, which from glutamate we can get glutamine, proline, arginine, as well as our purines. Purines are used to produce or to make DNA. Succinyl-CoA can be used to do porphyrins and produce heme. Pyruvate, we have a malic enzyme pathway that can convert pyruvate directly to malate and enter the citric acid cycle this way. And then oxaloacetate, a whole bunch of stuff can happen with oxaloacetate, where oxaloacetate can actually be converted back to phosphoenol pyruvate, and it can go backwards up to produce more glucose, or it can be used to produce a whole bunch of different amino acids, all these sort of stuff, all these sort of things. I would say with this chart, because there's a lot in this chart, and you will get asked a handful of questions from this chart. The big things I want you guys to remember is the malic enzyme pathway, glutamate, the fatty acids and sterols, and essentially the big idea is if you do not know and it's talking about this sort of thing, guess oxaloacetate, because 80% of the time you'll be correct with oxaloacetate. So the way that you'll be asked questions on this chart, you might see questions like we just saw before, or you'll see like what is the name of the enzyme that converts oxaloacetate to phosphoenol pyruvate. That's the PEP carboxykinase. Or sorry, PEP carboxylase is the one that does that. Nope. Sorry, I got confused in there's a lot in the chart. PEP carboxylase does the aspartate, asparagine, and pyrimidines. So, like I said, there's a lot here. Don't try and memorize all of this. Just get yourself a general idea kind of what's going on here. And those sort of questions will come from here. If you have the time, I'd recommend drawing this out as well a couple of times. Just like the big ideas of knowing alpha ketoglutarate, glutamates, and the purines, the pyruvates to malates, oxaloacetate pathway is a good one. He likes to ask about tyrosine and tryptophan, as well as aspartate and asparagine to pyrimidines. Those are two that I would say strike a chord in my brain. And then a little bit of the PEP carboxykinase as well. I recall that being something he would occasionally talk about as well. And he doesn't cover this a lot in the lecture. He kind of breezes through it, but he will pull a handful of questions off this. I'd recommend going through it. And in the book, it doesn't spend a lot of time on it either. It just sort of throws it in here like, oh, hey, by the way, there's this thing. But it's a good, he will pull a couple of questions off of this as well. Awesome. Thank you guys so much for your participation, and apologize for any of the little mistakes. Just remember that I guess it makes me feel a little bit better knowing that teachers are all human, that there's always going to be some mistakes, but I will try and make sure I get all of those fixed for you guys. And I will share this Kahoot out so you guys can practice it a bit. I'd also recommend, if you guys can, similar format questions like this, if you have the time, sit down and write some questions like this and write them out, or like put them into a Kahoot or something like this. The actual making of a Kahoot or of a Quizlet is a really, really good way to study, and making practice questions really forces you to look at the material closer, and will help you retain it a bit better as well. And congratulations to CC, Becca, and C being on our podium. Okay, does anybody have any other questions, concerns, anything like that for me before we wrap up?
No, it was great. It was great. Thank you.
Hopefully this gave you a little bit better idea. Hopefully this gave you a little bit better idea kind of what to focus on, kind of how to study, and hopefully gives you a little bit better idea of kind of maybe what you're lacking in, and need to spend some time reviewing, so your study can be a little bit more focused. Okay. Thank you guys so much for joining, for participating, and I hope you guys have a great rest of your day. Good luck.
Thank you. You too. Bye.